given the very special circumstance the entire world is in right now with COVID, can you talk about viruses? So what are viruses? How are they different
Yeah.
from normal critters? And how can understanding the ways that they evolve and the ways they evolve differently from other creatures, what can that tell us about how we should be handling the situation today?
That's a great question, Nick. And we're having this conversation right when I think the reports this morning or last night, where's this, new variant in Colorado. What does all this mean? Okay, so viruses are circulating everywhere. You and I've got them in us. That viruses are little bits of genetic material that replicate inside host cells. So these are not things that can exist free living out on their own, like say, for example, bacteria can, or fungi can. So they require a host cell to replicate. And animals harbor a lot of viruses. And if you think of all the diversity of animals on this planet, there are a lot of viruses across the globe. And every now and then, when there's contact between humans and wildlife, is an opportunity for viruses to jump from that wildlife into humans, an event often referred to as spillover. So the virus is spilling over from a natural reservoir into humans. That's what happened with COVID. That's what happened with HIV. That's what happens essentially every year with flu. It makes a cycle through usually birds.
Oh, really? I didn't know
So
that about the flu.
flu's recombining with viruses in pigs and chickens and other domesticated livestock to get
Okay. So
the
every
different variants
year,
of flu. Every year,
most years, it's a spillover event.
essentially it's a spillover event with flu.
Okay.
Yeah.
Wow.
So, but getting back to something like COVID, HIV is another good example, but it's of animal viruses coming into humans and then being transferred from human to human to human. So viruses, they are small, they are present in large numbers. When a virus replicates in you or I, it makes lots and lots and lots of copies of itself. So with lots and lots of copies of itself, there's lots of chance for evolution, because if you make 1 billion copies of a virus, those billion copies are going to carry a mutation here and there. And those mutant variants may have different properties than the parental virus. That's why, for example, this variant is making news, is because it looks like it might be a bit more transmissible than sort of the parent strain of coronavirus. And if it's more transmissible, what's that going to mean in terms of how it moves through the human population? Doesn't look to be more pathogenic, does not look like it makes any more severe or less severe illness. It just may be, for example, binding to our cells a little more readily. And so a little bit of virus gets a foothold more easily than the parental virus. So why is this all relevant to us? We live in a constantly evolving environment, and nearly 8 billion of us are making a lot of contact with wildlife that we didn't necessarily make before, through bushmeat or through encroaching in forests, as we cut down the Amazon, as we invade the Congo, et cetera, et cetera. That's where Ebola is coming from. So Ebola is coming from what we think is a bat host in its repeated cycles of infection, spilling over to humans. So, if you thought that evolution was just some subject that was for, dusty books and fossils and, old natural history museums, no, we live in a world where all sorts of things are evolving. The microbes that we try to treat with antibiotics are evolving. The viruses that are out there in the world are evolving. And as we've now experienced, these evolutionary events can have profound consequences for humanity. I can tell you that 4 years ago, we made a film on spillover and talked about Zika and Ebola and some other things like Nipah and flu. I never imagined something like this. I never imagined that a virus could spread so far, be so dangerous, and essentially paralyze human activity across the globe for a year or more now. I'm also glad that molecular biology has evolved very quickly, and that we could develop countermeasures like these vaccines, faster than any time in history. So, so from a perspective point of view, it's an accident. Random mutations happen in viruses, just as they happen in us. Some of those random mutations change those properties of the viruses, a chance encounter with a human, and we're off to the races.
So you mentioned how fast we've been able to adapt to this virus. We've been able to basically go from 0 to 100 in terms of vaccine development in a very short amount of time compared to historical vaccine development. What is different about how this new generation of vaccines from Moderna and BioNTech are working? And how does that impact our ability to try and to keep pace with viral evolution?
Yeah, great question. The way vaccines usually were made, say, let's say in the lowest tech era,
Mm-hmm.
were often just a killed form of the virus. So you'd grow the virus in vats of tissue culture in a lab. You'd inactivate it chemically or with ultraviolet or whatever it might be. And you inject that into people. So it's not able to infect them, but they're basically seeing the killed virus stimulates their immune system enough that they have protection in case they see the actual live virus. Those vaccines, which we use for lots of things, so flu is an inactivated vaccine, um, um, have generally worked okay. But for some viruses, they're very, very difficult.
Mm-hmm.
AIDS, we still don't have a vaccine because that virus mutates so quickly that basically there's no virus you could immunize people with that would protect everybody against AIDS. So, and usually because the way we conduct the clinical trials of these vaccines, there's a fair amount of sort of patient bureaucracy with this in that, you take one step and then it's sent off to the FDA and it takes months to review and then you start the next step. What happened was all those things were compressed. So basically the federal government stepped in and started funding development of the vaccine or the advanced stages of the vaccine before the other stages were sort of closed, so that these manufacturers could be scaling up the production of the vaccine before they even knew whether it worked. Otherwise, it'd be huge financial risk. If you start scaling up a vaccine before you know it works and you find out it doesn't work well, you may have lost $1 billion and your shareholders are very unhappy. So basically, the federal government backstopped a lot of these vaccine developers so that they could be scaling faster, so that they could jump into clinical trials faster. But there's also a technology shift. So instead of killed viruses, Pfizer and Moderna, or BioNTech and Moderna, use a technology that's never been used for large-scale vaccination, in that they are actually putting messenger RNA, which is a form of RNA that carries the genetic information for making one piece of the virus. They're putting that in little lipid droplets, injecting that into your arm, and your cells make a little bit of the virus that then primes your immune system. So these are mRNA vaccines, again, never been used at scale, and goodness, nobody could have predicted they would be as effective as the data turned out to be. Another thing about science, everybody's got to keep in mind, you don't know the results until you actually do the experiment. And for Moderna and BioNTech, Pfizer, it just turned out really well. So there was a technological change. And the thing about mRNA vaccine was that as soon as those viruses were sequenced in China from patients, we could in the lab here make those molecules very quickly and get them tested in animals and start right on the course of vaccination. So everything was sort of done in hyperdrive. Now, just today in the UK, a different vaccine made by Oxford AstraZeneca was approved. And this is, again, technologically something different, which is a little piece of the coronavirus, the same part that's being used in the mRNA vaccine, is in a vector, another virus, a harmless virus called adenovirus 2. And this is a virus that can't replicate in our cells, but acts as sort of a shuttle to get that genetic material into our cells, making a little bit of the virus so that our immune system gets primed to it. So it's ready to go in case we encounter the real virus. So both of those technologies are ways of delivering the sort of important part of the virus that is involved in infecting our cells. So this spike protein that's on the outside of the coronavirus, that's the sort of the key that unlocks the portal on our cells that gets the virus into our cells. And if you block that key, it can't get in. So that's the rationale for vaccine design. But researchers were working on coronavirus vaccines for things like SARS and MERS. They had some years of experience with these technologies, so that when this coronavirus came along, they said, hey, let's see if we can make a vaccine this way. So it wasn't like the research did not exist, but really it was benchtop research for other coronaviruses that they then said, well, let's go for it. Let's see if we can make an mRNA vaccine. Let's see if we can make an adenovirus-based vaccine. Another technology, which is just to make the spike protein as it is, that's the strategy being used by a company called Novavax. They're just entering phase 3 trials now. That's very similar in sort of design to something like the hepatitis B vaccine, which is pure viral protein. So that's a little later than these other vaccines. But this reflects, the biologist tools for doing these things have changed a lot over the decades. And so we had more tools that worked a lot faster so that lots of folks jumped in. I mean, this was a worldwide effort, and there's, lots of vaccines under development. But because of the societal imperative, the financial backstopping of these companies so that they could start making tens and tens of millions of doses before they even knew the vaccine worked. That was really important.
Mm-hmm. So I've heard the new mRNA vaccines described as programmable. What does that mean, and what would it allow us to do if, let's just say tomorrow, there's a brand new coronavirus strain, brand new mutation, the spike protein's now completely different, and the vaccines we have right now aren't as effective? How quickly could a new vaccine be developed?
Well, I mean, it's just going to be a speculation. I think it was 60 days for the first one that essentially the vaccine was formulated. So I think it's probably 60 days or less that we'd be able to spike. I shouldn't use the word spike. So right now, the mRNA that's in there is for essentially the prototypical strain of coronavirus. If we thought now that coronavirus had evolved enough that we wanted to have some variant or combination of variants represented in that vaccine, we could add the mRNA for those variant proteins to that mixture. Same technology. So I think that adding a variant to the Pfizer vaccine or the Moderna vaccine would be pretty darn straightforward. Now, I don't know, the regulatory authorities might want a new clinical trial to make sure that that was protective. Or it may be, again, just because of the imperative, because we're in the middle of a pandemic. Realize most studies are done at a slower pace because the urgency and the societal impact is not that great. That, maybe the regulatory authorities would allow use of that vaccine, essentially piggybacking on everything that was established about the first vaccine. They'd still want to probably gather data in the field, but they might allow the vaccine to go forward without all the steps to the trial. Now that essentially the principle has been proven, for the mRNA-based vaccines. So we would have a response, and the adenovirus is also programmable so that we could quickly be expressing that variant. So I think it's 60 days that we'd probably be working on it. Whether or not the regulatory authorities would allow that to go into people in large numbers without some formal testing, I can't speculate. But if this was something, imagine this: if 2 or 3 months from now we found out there was a vaccine-resistant strain, a strain that we didn't think was starting to circulate, that was not protected against by the vaccine. Man, I don't know. That's a really tough ethics call for regulators, which is, the world is really suffering. And would you take that chance? Well, I hope we don't have to find out, man. I hope we don't have to find out. But, we feel good that we can respond on this time scale. At the same time, every month going forward, you can see what the death toll and what the illness and the economic toll is going to be. So, every month really matters right now.
Yeah, it's amazing that the science is at a state where we can actually move this fast. It's absolutely amazing.
Yeah, it is amazing. And there's one other part which is probably unseen by the public. So they're getting the news reports that say, the phase 1 data that came out, I almost can't remember, probably midsummer, that said, look, when you put these vaccines into people, their immune responses are similar to people who got the real virus, got infected and went to the hospital. Then the efficacy data surprised everybody. I mean, no scientist is going to say, yeah, I'm going to make a 94, 95% effective vaccine out of the chute. A lot of the vaccines we have aren't 94, 95% effective at preventing infection. That's just the way the biology works. So, you know, they hit a grand slam first time at bat. That's you know, that's pretty remarkable. So I think, you know, the scientific community has a lot to be proud of, but the invisible part is that, you know, the sharing of data
Mm-hmm.
and the sharing of studies, because a lot of stuff is going to a lot of scientific studies of the coronavirus are going to these preprint servers before they go to journals. This is an innovation that happened in science publishing over the last few years to sort of speed up the otherwise somewhat slow pace of formal scientific publishing. Now coronavirus hits and people are just sharing their findings, you know, before peer review. There's some risk to that, but the upside of that is that the information is getting into each other's hands much, much faster.
Mm-hmm.
And man, I've tried to, you know, penetrate the mass of that literature when I go ask a question and say, well, what do we know about patient's response, you know, 3 weeks in? And I find all these papers from all over the world, and it's just happening at lightning speed. So the ability for scientists to share information is unprecedented. And the amount of sharing that's going on globally is unprecedented. Everyone's trying to learn as much about this virus and our responses to it and what works and what doesn't work as fast as possible and sharing that openly. That's a pretty remarkable thing. Sure, we'd love to know more. We all, you know, we wish we knew in March what we know now, and I'm sure we're going to know things this coming March that we wish we knew today. Look, but nonetheless, the sort of everything about the scientific enterprise is moving at a speed I've never seen in my lifetime on the coronavirus in particular.